Okay, folks, so in this presentation, we're going to be talking about the process of meiosis, which is a type of cell division that produces the reproductive cells of sexually reproducing organisms. And if you look at this diagram, you can see here that this cell is divided, and we end up with these four sex cells after each division. So before we get into meiosis, we want to just jump back to mitosis, which is the type of cell division we first looked at. And if we apply this type of cell division to a human, we have 46 chromosomes. And what happens is when we need new cells, or we need to repair our bodies, or we need to grow, we need to duplicate those cells. And at the end of that duplication, each of the new cells needs to have 46 chromosomes, because that's the genetic information that codes for us. So in the case of mitosis, we have interphase, and we have interphase in meiosis too. But when the nucleus divides, that's when we see actually mitosis. And mitosis is prophase, metaphase, anaphase, and telophase. And these stages just describe to you where the chromosomes are as the nucleus divides. Finally, the cytoplasm divides, and that's cytokinesis. So some new terms we're going to see when we talk about meiosis are diploid, haploid, tetrad, and homologous. Each of these terms is going to reoccur, so we want to start to get a handle on them now, and you'll see them, so take note as we go through the PowerPoint. So we're going to start this off by talking about these things called homologous chromosomes. We want to define and describe the structure of these chromosomes. So homologous chromosomes, or chromosome number, has a lot to do with this whole process. We're going to be keeping track of the number of chromosomes in the product of mitosis versus meiosis. And in all living organisms that reproduce sexually, they get a complete set of chromosomes from each parent. So if we look at the fruit fly, a fruit fly has 8 total chromosomes. However, it gets half of those, 4 from the male parent and 4 from the female parent. So if we look over here at this diagram, this is actually what's called a karyotype. And a karyotype is simply chromosomes laid out in size order. So continuing with the karyotype, the 2 sets of chromosomes from each parent are considered homologous. So each chromosome that comes from a male parent has an exact match from the female parent. So if we look over at the karyotype again, we see here's a set of homologous chromosomes. They're similar in size, and they carry similar genetic information. And try not to get these confused with sister chromatids, which are really just these ends here, and these are just duplicated during cell division. But the homologous chromosomes are a little different. They're from mom and dad, and they are a set from each parent. And down here you can see the sex chromosomes, which determine whether you are male or female. Oh, and by the way, if you look at all these chromosomes, this is a human. If you were to count them, you would find 46 total, and each of these would actually make 23 pairs. So cell types. We really are looking at the sets of DNA when we talk about cell types. So cells with both sets of chromosomes are called diploid. Di meaning like dice, 2. Diploid cells are the body cells. So if we look down, say this is the body cell of a human, and we're looking at, let's say, a skin cell. In a human, they don't show them all here, there would be 46 chromosomes in that initial skin cell. And when that cell divides through mitosis, we end up with 2 new cells. Each of those has those 46 chromosomes. And you can see that original set from the beginning is still there with those 2 homologous pairs. Remember, we have 1 from mom, 1 from dad. And we often represent this by writing 2N, meaning 2 sets of chromosomes, diploid. On the other hand, when we talk about meiosis, meiosis actually produces cells with 1 set of chromosomes, and these are called haploid, which kind of means like half. And if we haploid cells are sex cells. So here's our sperm produced by meiosis. Also, eggs are produced this way. So meiosis produces cells with half the genetic information of that parent or that organism that made them. Meiosis, on the other hand, produces chromosomes, cells with two sets of chromosomes. So this would be 46 in the case of a human. Um, and that's more for growth and repair of the organism. So that's more what mitosis is for. Meiosis, just going back, produces these sex cells with 23 sets of chromosomes. And eventually they'll meet up with an egg, and then they will restore that 46 chromosomes in the new human that gets created after fertilization. So going a little bit further, meiosis forms gametes, and gametes are genetically unique haploid reproductive cells, such as sperm or egg. We take one diploid cell in meiosis and we divide it a few times until we end up with this haploid cell, and it's genetically unique from the parent and also the other sex cells that are produced with it. So once again, diploid cells are a result of mitosis. This is more for growth and repair, so we're looking at growth here when you grow from a child to an adult, and also repair. If you get hurt, you are going to grow new cells to fix your body and maintain it. On the other hand, haploid cells are produced by meiosis, and those are like sperm and egg. In the case of humans, those cells have 23 chromosomes because we actually have that set we have in all of our other cells. So moving on, we want to describe how meiosis, sexual reproduction, leads to the development of haploid cells and gametes from diploid cells. So we want to describe this process. So like mitosis, meiosis has different phases. So we want to describe those and what's happening to get a better idea why the result that is created is made, which is these sperm and egg cells. So what is meiosis? Meiosis allows for sexual reproduction. The number of chromosomes per cell is cut in half by by separating chromosomes in a full cell. So in this case, if we start with a human, we have 46 chromosomes in our cells. Every cell in your body has these except for your sex cells. And we have these homologous pairs of chromosomes in it. There's one there, those two that are similar size, and we have this other pair here. Notice they're different colors. That's supposed to mean one comes from your mother's side, one comes from the father's side. And through this process of meiosis, we end up with these 4 cells rather than 2 like in mitosis, and they are actually genetically unique, and they have half the genetic information. So each of these has 23 chromosomes rather than the 46 that would be there if this was mitosis. So meiosis, we're taking 46 chromosomes and we're separating those in half. And there's actually some genetic mixing and shuffling that goes on in here too, which is part of the reason you're not identical to your brother or sister, unless you're an identical twin, and you also don't look exactly like your parents. So just to reiterate that, 23 chromosomes in a haploid egg cell, 23 chromosomes in a haploid sperm cell. Those meet during fertilization to form a zygote, and when that zygote develops, mitosis kicks in after this point, and all those cells are dividing those 46 chromosomes up to make all the cells, the trillions of cells that make you. So pretty amazing. Be impressed. Smiley face. So this is just another idea where if we didn't have this reduction division or this splitting in half of those 46 chromosomes, we would end up producing children with double that amount of chromosomes and so on. And that would really, the amount of DNA we have is what's best. So we don't want extra chromosomes or extra DNA. So meiosis is important for cutting that number down, 23 and 23, and combining those to make 46 again in the new organism. So this way every adult human, unless you have a genetic disorder, has 46 chromosomes. And here's another example of that, the problem. We don't have that reduction division, no meiosis, we end up with this doubling of chromosomes, and at that point you no longer have a human, you have something else. So the phases of meiosis. So there are two divisions required to complete meiosis. Meiosis 1 is the first. So the chromosomes are replicated in the S phase of interphase. So yes, meiosis still has an interphase. And then we get into the part where the nucleus divides. And that's going to be the first thing that happens is prophase 1. And this is a little different from mitosis. So each chromosome pairs with its homologous match, forming a tetrad. And a tetrad is really this, what you see right here. It's the chromosomes kind of getting together and crossing up a little bit. And what happens now is crossing over takes place, which is the step where genetic information is exchanged between the chromatids. So now what's happening, not only are we dividing the cell, we are mixing up the DNA a little bit. So then what? The homologous chromosomes separate by the spindle fiber, so we're still pulling chromosomes apart. The two new cells are formed, and the cells are genetically different from each other, from the original parent. So this is new. Remember, in mitosis, we see an identical clone cell being created. However, in meiosis, all of these cells that are produced are going to be different. So remember that. But right now we're only focusing on meiosis 1. So inside this circle is meiosis 1. And meiosis 1 includes a prophase 1, a metaphase 1, an anaphase 1, and a telophase 1 as well. And we end up with these two cells that are genetically different. And notice at this point, they're trying to show you the colors are a little mix and match here between the homologous chromosomes, and what they're showing you is that they've crossed over. They've exchanged genetic information to become slightly unique from the parent. So once again, that's part of the reason we don't look just like our parents. We look kind of like them, but a little different. And you can see the homologous pairs are separated, but our sister chromatids are still there. So see, there's a sister chromatid, there's a sister chromatid, there's one, and there's one. So the sister chromatids are still together after meiosis 1, which is different from mitosis. So just to look at that again, here's our interphase. DNA and organelles are being replicated, getting ready for division. Prophase 1 occurs. We have crossing over between the homologous chromosomes. Genetic information gets mixed. And then we have our metaphase, anaphase, and telophase. And instead, during anaphase, see how the whole homologous chromosome is being pulled apart here? So we're pulling the homologous chromosomes apart, but we're not pulling the chromatids apart. So you can see the sister chromatids are still going to be intact at this point. So meiosis 2, kind of the sequel to meiosis 1, and that's what's going to finish this whole thing. The two cells from meiosis 1 then enter a second division. No chromosomes replicate, and in telophase 2, 4 daughter cells are produced, and each has half the genetic information, so they are haploid. And by half the genetic information, remember, we started with 46. Each of these has 23 chromosomes. So we went from those 46 chromosomes in the original cell in a human to 23 chromosomes. And meiosis 2 occurs again with, it has a prophase, a metaphase, an anaphase, and a telophase, except it's a little different. We're actually splitting all of that genetic information up, the chromosomes, into 4 new cells with half of the original chromosomes. So looking at meiosis 2 a little more closely, you can see here, now we're splitting apart those sister chromatids finally. And notice the exchange here. Remember, they formed tetrads and crossing over happened. So now these chromosomes are different from the original parents. So this is part of the reason you're different from your parents a little bit. So we have a prophase 2, metaphase 2, anaphase 2, and telophase 2. And now we have these final cells. And remember, these all have 23 chromosomes instead of that original 46. And they're all genetically different from each other. So unlike mitosis, these are all different. And this is the reason that we all look different from our siblings and our parents. Unless you're an identical twin, you look just like your sibling. I'm going to skip that. And just to summarize, when sperm cells are made, because remember meiosis makes sex cells, we call this spermatogenesis. When egg cells are made, we call this oogenesis. And remember, we have those two divisions. Both of those have those divisions, both processes. These are both meiosis. And in the case of a male, we get these 4 sperm. Remember, we had our 46 chromosomes here. We have 23 in each of these. And in the case of a female, it's a little different. We do get those 4 cells, but only one is viable. So we only get one viable egg. Also 23 chromosomes and genetically different, thanks to crossing over. So remember, crossing over occurs in meiosis 1. You might want to put a little note to yourself. So crossing over occurs here. And crossing over just means genetic mixing. Or variation is what's made out of that, really. And all sexually reproducing organisms, not just humans, have crossing over. So we've had the number of chromosomes, we've had 2 divisions, meiosis 1, meiosis 2, and we end up with these sex cells. So just want to get into crossing over a little bit more and explain how crossing over leads to variation. Variation is the key. The fact that we're all varied is a very important part of biology, which we'll study more, especially when we get to evolution and natural selection. So looking at crossing over, remember the tetrad? This forms during prophase 1. You want to remember that? So it's in the first part of meiosis. And this tetrad, you can see they've done a little dance here, and they've kind of exchanged pieces, these homologous chromosomes. Remember, one from mom, one from dad. They're similar in size, but they kind of mix the DNA up a little bit. So this is part of the reason you look similar to your parents, but your traits are a little bit different, or they're mixed up in a way, and this is part of the reason for that. And then the process continues through each division. So we have metaphase 1, anaphase 1, metaphase 2, anaphase 2, because we have those 2 meiotic divisions. And we end up with these 4 genetically unique haploid sex cells. Because remember, even though we're only seeing 2 chromosomes here, we actually have 46 in a human. And all of these are going to have 23, so they're going to be haploid. And we call this random assortment of genes. So what happens is the new genes, or the new gene combinations that we make in these chromosomes, are going to be randomly assorted into the new sex cells. So every sperm cell is different. It has different genetic information. This is why brothers always look a little different from each other unless they're identical twins. And the same with egg cells as well. So we don't know what's going to happen with those traits exactly. They're randomly assorted into the sex cells during the division process of meiosis. So just a closer look at that, I'll go through this quick. We have, if we want to catch this in motion, we have the homologous chromosomes. They line up, they exchange some genetic information in that tetrad, and then they have exchanged that information. You can see here we have the exchange. That's what the colors are trying to show you. So now these are genetically different and unique from the parent. So meiosis problems. Occasionally some problems occur during meiosis. These are called mutations, and they are changes in the DNA that can occur because of crossing over. The DNA can change in the process of meiosis, and when chromosomes replicate. So basically, sometimes when the chromosomes replicate, remember we have those sister chromatids. Sometimes when they're supposed to split and each go their own separate ways, we have this problem where they only go one way. And we end up with too many chromosomes in one organism, or in one of the sex cells. So we'll talk more about the implications of this in the next slides. So non-disjunction. So non-disjunction was what I was just talking about, where we don't have that complete separation of the chromosomes. So in this, we're just going to explain how non-disjunction causes various genetic disorders. We're going to talk about 2, Klinefelter syndrome and Down syndrome. So meiosis problems. Non-disjunction occurs when anaphase is not completed. Chromosomes are not properly divided, and the gamete ends up with an extra chromosome. So if we look at meiosis here, here's meiosis 1. A little problem happened here. these chromosomes were supposed to go their separate ways. However, they ended up all going one way. And so they both ended up in this cell after meiosis 1. So the process continued, and now we have these cells down here that ended up with too many chromosomes. And that's going to make for some problems for our organism. So just looking at these, another show of that, and what we're seeing here is in A, we have our normal meiosis. We have our chromosomes here, 46 chromosomes, we'll say, it's a human. They divide once in meiosis 1, so we'll call that M1. And then they divide again through prophase, metaphase, anaphase, telophase in meiosis 2, so we'll put M2 here, Roman numeral. And we end up with these haploid cells with the proper amount of chromosomes or genetic information in them. Each has 23, so we'll call those haploid because they don't have that original diploid number. And here's, just to remind you, this is diploid up here because there are 2 sets of chromosomes in the original cell. So now, if we want to see non-disjunction, it can occur in 2 places during meiosis. It can occur during meiosis 1. So during M1 here, we had, or meiosis 1, we had a little problem where too many of the homologous chromosomes ended up in one of the cells produced by meiosis 1, and the other didn't quite get the amount it was supposed to. So we end up with a non-disjunction event. They didn't disconnect and they stayed together, and these are the homologous chromosomes. Remember, in meiosis 1 the homologous chromosomes separate. You can still see the sister chromatids there, those 2 ends, and here's the sister chromatids here a little more clear. So that's B, and we end up finally with cells with too much genetic information. So this is usually a pretty major problem for a person. Many times people who have big time chromosome issues like this are not able to survive birth. C, so we look at C, we have our meiosis 1 and meiosis 2, and in the example here we have the sister chromatids, and meiosis 1 goes as planned. We have those chromosomes, those homologous chromosomes separate as they should during anaphase, but in meiosis 2 there's a little problem. In anaphase 2, these chromosomes didn't separate, or the sister chromatids didn't separate like they should have. We should have just 1 in here, like the original drawing. So in this case, we have an extra chromosome in this cell, and that's going to make for genetic disorders depending on which chromosome it is. Because remember, we have 46. So one disorder that can be caused is called, by non-disjunction, is called Klinefelter syndrome. And in Klinefelter syndrome, we got an extra X chromosome ending up in the sex cell that's produced by meiosis. Normally, boys and girls have just 2, because remember we have 2 parents. And so in this case, a girl would have 2 X chromosomes, and a boy would have an X and a Y chromosome. If we look at the karyotype here, someone with Klinefelter's ends up with 2 X and 1 Y. So this is going to make for some different physical features for them. Here's our normal male here, and you can see someone with Klinefelter's is a little bit different. They have the extra chromosomes, so they have 47 chromosomes total, and this person would look like a boy except a little more feminine, broader hips, and a more slender body, narrower shoulders, and usually these people are treated with hormone treatments to help give them more the figure of a normal male. Another syndrome caused by non-disjunction is called Down syndrome. And Down syndrome is a situation where the sex chromosomes are fine, however, the 21st chromosome, and we have a non-disjunction event and end up with an extra 21st chromosome there. So now a person with Down syndrome actually has 47 chromosomes because they have this extra, and that can make for some different physical features and other traits as well that are different from someone who doesn't have the extra chromosome. And just an interesting note, this person is a girl, and you can see they have 2 X chromosomes and no Y. So fertilization, we want to explain how fertilization restores the number of chromosomes in a species and ensures the inheritance of maternal and paternal characteristics from one generation to the next. So that's a lot of words. Really what it means is our mom has 23 chromosomes with her unique DNA in her sex cell, and our paternal, our father, has 23 chromosomes in his sex cells because they were made in meiosis and they're genetically unique from him and each other. And those characteristics from them are going to be put into the new generation, meaning the new baby, and that person is going to be unique. So that's fertilization is really the cells produced in meiosis doing the biological job they were meant to do. So what's going on here? I think we all kind of know. In general, sperm will come in contact with an egg, and only one of these guys will make it. I'm going to say this one. And once they unite, we have 23 chromosomes, remember, in the egg. We have 23 chromosomes in the sperm. They unite to create a new person, 46 chromosomes, and then we have a lot of mitosis going on to make a lot more diploid cells to grow that organism or person into an adult eventually. So fertilization is the fusion of sperm and egg to form zygote. A zygote is just that first cell that's produced after fertilization. So just to summarize, we have our egg and sperm meeting during fertilization, and they produce a new zygote. And really, this zygote is going to be a baby. Let's see if I can draw a baby here. So that baby will form from the zygote eventually. Not bad, look at that. And they will have 46 chromosomes. Then we're going to get a lot of mitosis going on to grow that person. They won't be doing much meiosis until they hit puberty, or at least if they're a male. So gamete formation, the cells develop by meiosis, form sperm cells, and females produce egg cells. So male animals, let's focus on them, and we saw this a couple slides back. We start with this original diploid cell. Meiosis 1 occurs. We have all those first phases to divide that nucleus, except remember, we're just separating the homologous chromosomes. The sister chromatids are still intact, so they separate. Then we have meiosis 2, and in meiosis 2, we end up with these 4 haploid cells. And in the case of a person, 2N means 46 chromosomes, and just N, all these sperm have 23. And remember, all of these sperm that are produced are genetically different from each other, and they're different from the parent. So that's why, unless you're an identical twin, you look different from your siblings and your parents. Well, you look like them, but your traits are kind of rearranged, and that's because of crossing over, remember, in prophase 1, where the homologous chromosomes exchange genetic information. So female animals, on the other hand, one of the 4 cells produced in meiosis receives most of the cytoplasm, and the other 3 cells generally do not participate in reproduction, as they do not have the necessary structures. So what that means is it's a little different. Oogenesis, or meiosis in females, the egg-producing process, same idea. We start with that 46 in the diploid cell, and we go down and we end up with the 23 in one egg cell. We might say, well, aren't there supposed to be 4 products of meiosis? Well, there still are, but these other things are called polar bodies, and the polar bodies are going to contribute their nutrients to the one viable egg, or the egg that's going to be used. And these polar bodies will dissolve and go away. So an egg needs a lot of energy, because remember, the sperm are lightweight, they're traveling. The egg is going to be used to kind of kickstart the whole growth of that zygote, which is going to grow into a baby. So we need to kind of get all the nutrients we can into that one egg to help that baby grow. Through mitosis, remember. So fertilization occurs when 2 gamete cells are united and differentiation may begin. By differentiation, we mean cells start dividing and eventually new organs are formed and different types of cells to make the organism. So a sperm cell meets an egg cell, we get a diploid offspring, so diploid. And that offspring or baby, remember, one thing to keep in mind is we've been talking about humans, but I mean, ferns do this, flowers do this. They have their own, and flies, they have their own number of diploid and haploid sets of chromosomes. But the bottom line is when they meet, we have those single sets. In the case of human, it's 23 and 23. They meet, we get the new diploid organism 46. And then we get a lot of mitosis, making lots of new cells to grow that new organism or baby. So mitosis after that, and before, to make these cells, we have meiosis. It's hard to write with a mouse. Mitosis can begin after this. So here's a electron microscope picture of sperm meeting an egg. And remember, only one sperm can get into the egg. After that, the egg, as soon as that first one reaches the egg, all the others are instantaneously locked out. So meiosis, the whole point is, we lay it out here, results may vary. Meiosis creates these haploid varied cells. So that's what's so important about it. It is what makes all of us different genetically. This is why we all have different heights, different traits, different interests. A lot of that isn't just because of our social or our socialization. It has a lot to do with our DNA. And remember, just to summarize, we have prophase 1. Remember, that's crossing over. So crossing over is where we have that mixing of DNA. So don't forget crossing over, very important, occurs in prophase 1. And we get that first division. Remember, we start with 46 chromosomes in a human, and by the time we get here, we have 23. So we have M1 here, meiosis 1, and then we have meiosis 2, meiosis 2 occurring down here. And you can see how they show those little exchanges of color. That's because crossing over occurred. Notice in meiosis 1 as well, we'll jump back, we're splitting the homologous chromosomes, and in meiosis 2, we're splitting those sister chromatids to end up with these haploid cells with just one half of the genetic information from the original parent. And all of these are different from and varied from the parents. So we get this genetic variation from the parents and from siblings, and in general the entire human population. So mitosis, or meiosis versus mitosis, we want to compare and contrast these. They're very similar, but they serve very different purposes. So mitosis begins with one diploid cell and ends with two identical diploid daughter cells. So keep that in mind. We start with a diploid, we end with a diploid. So we have these chromosomes here, they get duplicated. There's the sister chromatids. Those sister chromatids get pulled apart, and we end up with these two diploid cells again. So, and they are clones. Remember, these are clones of the original cell. They're exactly the same. 46 chromosomes in a human, and we have 46 in each of the new cells as well. It occurs in somatic cells. Somatic just means body cells such as liver, heart, skin, anything but the sex cells, any other cell. And this allows cells to grow and reproduce. So when your body needs to be repaired, or we need to add some new cells for growth, we use mitosis. And this allows for asexual reproduction, meaning exactly what this is, 46 chromosomes, and we're creating clones, and we're ending with 46 chromosomes, diploid to diploid. And this happens in the body cells of an organism. Meiosis, on the other hand, begins with a diploid cell, just like mitosis. However, we get 4 genetically different haploid cells. So this allows organisms to reproduce sexually, and by sexually, I mean organisms are able to actually exchange DNA in a way. When your parents make sex cells and those sex cells unite, they make a unique person, which is you. And this is different from mitosis, because in mitosis, if we were doing that, we would all be exact copies of our parents, or at least one of them. So in meiosis, a little bit different. We have meiosis 1, so we have 2 rounds of division, and we have meiosis 2 occurring to give us these 4 cells. And remember, we start with 46. We still have that duplication. There's the sister chromatids, but in this case, those homologous chromosomes get pulled apart in the first division, meiosis 1, and in meiosis 2, we have the entire, or we have the sister chromatids getting pulled apart to make those genetically unique haploid cells with 23 chromosomes in the case of humans. And remember, up here we also have crossing over occurring in prophase, and that's what's mixing up and making the genetic information different by the time we get to the end. So comparing these two, remember, we start with a diploid cell in both. We have two identical cells formed in mitosis, and we have two different cells formed, or four different cells formed in meiosis. They're also haploid, where here they're diploid. This occurs in body cells, where this meiosis only occurs and makes sex cells. And when we have asexual reproduction, here we have sexual reproduction. So make sure you get down those differences. And looking at the two diagrams together, remember we start with 46 here. We end up with 46 here and here. So our cells that are produced are still diploid and exactly the same, asexually reproduced. In the case of meiosis, we have two divisions. We have crossing over, I'm going to write CO. So we have crossing over here in prophase 1, P1, and then we have another division, and we end up with these different cells. So remember, different, these are different. And by different, I just mean genetically varied. Where in mitosis, they're all the same. So those are really the things you want to remember. We still start with a diploid cell in each case. Mitosis, one round of division, 46 chromosomes, we make clones. Meiosis, 2 rounds of division, we exchange genetic information, change it up, and end up with half that information in each of the cells produced. Remember, 23 in each, and they are genetically different, and these are the same. We'll just put that in there. And here are our objectives. Make sure you get familiar. These are all the pieces that this whole entire PowerPoint went through. A lot of information in there. Make sure you jump around and take your time. A lot of information could get very boring too, so be careful. And if we just look at this process, you can use this too. Remember, the notes are online, and so are the practice packets. You can compare and contrast a little further here. So I'm not going to go through all these because it's going to add a lot of time, but I would do this on your own with the PowerPoint that's online. You can look at each, and it separates and shows each of the purposes of mitosis versus meiosis. So I hope this was helpful. You can record and reuse it, and hopefully as we go I will improve my skills. So long.